Cell cycle, mitosis, and meiosis. The cell cycle is divided into two main phases, inner phase and the M phase. Inner phase takes up most of the cell cycle and is composed of three substages, the G1 phase, the S phase, and the G2 phase. The G1 phase is the phase where the cell grows and develops. The S phase is when DNA synthesis occurs, which is when the cell duplicates its DNA. And G2 phase is when the cell continues to grow but is also preparing for mitosis in the M phase. Internal and external signals. Internal and external signals are used to regulate the stop and go signals at the checkpoints of the cell cycle. Important internal signals that are used to pace the cell cycle are cyclins and cyclin-dependent kinases. An example of a cyclin-dependent kinase, or CDK, is MPF, which is responsible for getting the cell from the G2 phase to the M phase, which is shown here. As you can see, the MPF activity goes up during the G2 phase and also is at its highest during the M phase. Mitosis and cytokinesis. Mitosis is the division of the nucleus, and cytokinesis is the division of the cytoplasm, and both of these occur during the M phase. Now, mitosis always starts with a parent cell that's diploid and always ends with two daughter cells with identical DNA that's identical to the parent cell and is also diploid. So in prophase, the DNA is in its chromosome form and the nucleus is gone. In metaphase, the chromosomes line up in the middle, and at anaphase, the chromatids are separated and move to opposite sides of the cell. In telophase, the nuclei form around the chromatids, and then the cytoplasm splits during cytokinesis. Meiosis is the production of gametes, or sex cells. Meiosis is always going to start with a diploid parent cell, and it's going to end with 4 haploid daughter cells that have non-identical genetic material to that of the parent cell, and this is due to crossing over. And crossing over happens during prophase 1 of meiosis, and this is when the chromosomes actually switch genes with each other, which creates more genetic variation. So the parent cell replicates its DNA and it divides into 2 daughter cells. And these 2 daughter cells are going to be haploid. And then these 2 haploid cells are going to divide, each of them are going to divide into 2 more haploid cells, which in the end results into 4 daughter cells that are haploid and have non-identical genetic material due to crossing over. Now this is how sperm is created through spermatogenesis. However, eggs are also created this way, except for only one is an actual egg. The rest are going to be polar bodies. Cancer and disruptions in the cell cycle. Cancer is due to many different things, one of them being that cancer cells are able to stop dividing and start dividing at random points in the cell cycle, regardless of the different checkpoints. Another thing about cancer cells is that they're able to grow and reproduce without growth factors, which cells need in order to reproduce. With that being said, they essentially are sometimes always they're able to divide forever. And another thing about cancer cells, which is shown here, is that they lack something called density-dependent inhibition. And density-dependent inhibition is when crowded cells stop dividing. Now, this is normal amongst regular mammal cells, but in cancer cells, since there's no density-dependent inhibition, they just pile up on top of each other like this.